Hey there, welcome to our video. Today, we're going to take a closer look at a research paper that explores the connection between familial Alzheimer's disease and small vessel disease. So, let's start with the basics. Alzheimer's disease is a common form of dementia, and familial Alzheimer's disease is a rare genetic form that accounts for about 5% of all cases. This study focuses on a specific mutation in the PSEN1 gene, known as the p.glu280 mutation, which is the most common cause of familial Alzheimer's disease. The researchers compared brain tissues from individuals with familial Alzheimer's disease, late-onset Alzheimer's disease, and a condition called CADASIL, which is a type of small vessel disease. They used advanced imaging techniques and histopathological methods to assess the extent of small vessel disease in these different groups. What they found was really interesting. Even though the individuals with familial Alzheimer's disease didn't show obvious signs of small vessel disease on MRI scans, the post-mortem analysis revealed that they did have significant small vessel disease pathology in their brains. This suggests that small vessel disease may play a role in the progression of Alzheimer's disease, even in cases where it's not immediately apparent. The study also highlighted the importance of early recognition and active management of vascular disease risk factors, which could potentially help prevent or slow down the progression of Alzheimer's disease, especially in familial cases. Overall, this research sheds light on the complex interplay between genetic factors, vascular health, and neurodegenerative diseases like Alzheimer's. It opens up new avenues for further investigation and potential therapeutic strategies. Abstract. We studied small vessel disease, SVD, pathology in familial Alzheimer's disease, FAD, subjects carrying the presenilin-1, PSEN1, p.glu280 mutation in comparison to those with sporadic Alzheimer's disease, SAD, as a positive control for Alzheimer's pathology and cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy, CADASIL, bearing different Notch3 mutations, as positive controls for SVD pathology. Upon magnetic resonance imaging, MRI, in life, some FAD showed mild white matter hyperintensities and no further radiologic evidence of SVD. In post-mortem studies, total SVD pathology in cortical areas and basal ganglia was similar in PSEN1 FAD and CADASIL subjects, except for the feature of arteriosclerosis, which was higher in CADASIL subjects than in PSEN1 FAD subjects. Further, only a few SAD subjects showed a similar degree of SVD pathology as observed in CADASIL. Furthermore, we found significantly enlarged perivascular spaces in vessels devoid of cerebral amyloid angiopathy in FAD compared with SAD and CADASIL subjects. As expected, there was greater fibrinogen-positive perivascular reactivity in CADASIL but similar reactivity in PSEN1 FAD and SAD groups. Fibrinogen immunoreactivity correlated with onset age in the PSEN1 FAD cases, suggesting increased vascular permeability may contribute to cognitive decline. Additionally, we found reduced perivascular expression of PDGFR-beta AQP4 in microvessels with enlarged PVS in PSEN1 FAD cases. We demonstrate that there is A-beta-independent SVD pathology in PSEN1 FAD, that was marginally lower than that in CADASIL subjects although not evident by MRI. These observations suggest the presence of covert SVD even in PSEN1, contributing to disease progression. As is the case in SAD, these consequences may be preventable by early recognition and actively controlling vascular disease risk, even in familial forms of dementia. Introduction. Alzheimer's disease, AD, is the most prevalent neurodegenerative dementia affecting approximately 46 million people worldwide. Familial Alzheimer's disease, FAD, accounts for up to 5% of all AD cases, caused by mutations in the amyloid precursor protein, AP, or presenilins 1 and 2, PSEN1/2, genes. The p.glu280 mutation in PSEN1 is the most common known cause of FAD with more than 600 symptomatic carriers in the Colombian kindred alone and presents with neuropsychological symptoms such as memory and language impairment and behavioral changes. We have recently shown multiple factors are responsible in the age of onset heterogeneity in FAD caused by mutations in PSEN1 gene. Other factors including pre-existing vascular disease should be considered whether they modify severity or disease progression. Accumulating evidence suggests that vascular factors modify disease onset proportional to the severity and the threshold for dementia. We have also detected individuals in the same region of Colombia with cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy, CADASIL, caused by two different point mutations, CIS455 ARG and ARG1031 CIS, in the Notch3 gene. Notch3 is a substrate of the secretase which presenilin-1 is the catalytic unit. CADASIL is a cerebral small vessel disease, SVD, characterized by the presence of subcortical strokes, microbleeds, and white matter hyperintensities, WMHs, on magnetic resonance imaging, MRI, that leads to vascular dementia. CADASIL shows several key features of SVD involving arteriopathy and small, perforating vessels leading to subcortical infarcts, microinfarcts, WM changes as well as perivascular spacing, PVS, and cerebral atrophy, 9. PV being suggested as a significant feature of radiologically defined SVD and is thought to result in periarteriolar obstruction of fluid transport and clearance of waste products by the accumulation of proteins or cell debris. Lacunar infarcts and arterial disease were previously reported in late-onset AD patients that showed no overt evidence of vascular involvement. The neurovascular unit, NVU, is comprised of different cell types, for example, endothelial cells, parasites, and astrocytic end feet. These control blood-brain barrier, BBB, function and they were shown to be disrupted late-onset AD. In apolipoprotein E, APO, epsilon-4 carriers, BBB breakdown contributes to cognitive decline independent of A-beta pathology. Therefore, the study of SVD in FAD patients provides a unique opportunity to assess vascular pathology without confounding factors associated with aging and common in the late-onset variants with usually several comorbidities. WMH of vascular origin, frequently used as the clinical signature for early detection and progression of SVD, are common in late-onset AD just as they area key diagnostic features of CADASIL. Current observations from the dominantly inherited Alzheimer network, DIEN, have described that FAD mutation carriers have greater total WMH volumes and white matter, WM, degeneration, which appear to increase several years prior to expected symptom onset. WMHs have a propensity for a posterior distribution and are largely attributed to cerebral amyloid angiopathy, CAA, which tends to be similarly distributed. These findings collectively suggest WMHs are a core feature of SVD in AD and a target for potential therapeutic strategy. Here, we assessed the extent of SVD pathology in a large FAD cohort of PSEN1 FAD. We hypothesized that clinical FAD may be modified by covert cerebral microvascular pathology. We compared previously established features of SVD pathology in FAD with that in SAD and in a large cohort of CADASIL subjects from two centers. CADASIL is an ideal model for SVD and microvascular pathology, largely free of AD or apathology. Further, with Notch3 being cleared by the secretase, the rationale to compare FAD-TO CADASIL is given because the one pathway is affected by the mutations. Quantitative histopathological and immunohistochemical methods were undertaken to evaluate SVD in a total of 88 subjects including brain tissues from normal aging old and young control subjects. So, there you have it, a fascinating glimpse into the world of Alzheimer's disease and small vessel disease. If you want to learn more about this groundbreaking research, be sure to check out the full paper. Thanks for watching, and stay curious.
Pharmapal. Pharmapal.